Good day, dear colleagues. I welcome you on our webinar, which would be focused on the research and synthesis of novel and generic drugs, including anti-infective drugs. So we will cover several examples, including development of the drugs against COVID-19 and other drugs, other diseases like hepatitis, HIV, and mainly we will talk today about the drug repositioning, and we will discuss several approaches which are applicable for the drug repositioning. The ideas and the approach which we will discuss today is also applicable to other diseases and other types of the drugs. For example, antineoplastic drugs, cardiovascular drugs, antidiabetic drugs, and the data-driven approach about which we will discuss today would be based on the clinical data from Embase and experimental data from the database Reaxys. Both of these databases available right now till the end of July to all Egyptian researchers via the Egyptian Knowledge Bank, via the platform, and my colleague Khalid Shalan will explain a little bit how you can access Reaxys or Embase using these platforms, and we will continue with the discussion. Again, I want to introduce myself. My name is Andrei Kudoshin. I have PhD in chemistry, and last 5 years I supporting customers around the world in developing the best technologies in drug research and development. And let's give the word to my colleague Shalan. Khalid.
Thank you, Andrei. I'd like to welcome you all here to this webinar. My name is Khalid Shalan. I'm an Elsevier consultant responsible for core research solutions. Since I'm based in Egypt, I'll be helping my colleagues, Dr. Andrei and Dr. Alexei, with the presentation to demonstrate how you can access the solutions that will be part of the topic for today using EKB platform. As you already know, EKB is only accessible from within Egypt. So I'll be showing to you, since I'm based in Cairo, this is why I'll be showing to you myself how to access the solutions from within EKB. So if you're able to see my screen now, so basically to start with the Egyptian Knowledge Bank platform, as you have registered to join us on this webinar. The EKB is one of the world's, if not the world's largest digital libraries. It is available on ekb.eg, Egyptian Knowledge Bank, the acronym for this one, .eg. The website is accessible only from Egypt, and the idea was to enable a community that thinks, learns, and innovates. It's about providing access to different knowledge resources to all Egyptians. So any Egyptian citizen with a national ID can get access to premium quality content provided by more than 35 publishers and service providers across the entire world. So basically, just as an overview for EKB, it includes 4 sub-portals. The first sub-portal is the reader portal, then the academic portal, basic education, and kids. Kids provides the resources to children. Usually we're talking about 6 to 14 years old. Basic education, it's integrated with primary and secondary education in Egypt. So it corresponds to the work done by the Ministry of Education. The ones that will be of interest to us for the sake of our resources as Elsevier resources are the readers and academic portals. Reader portal, that's the most generic portal. It means any Egyptian citizen with a national ID gets access to a very good range of content and services available to by the EKB from different publishers. They have all the general resources and latest news in areas of general interest. Uh, by design, the academic portal is the most premium portal on EKB. It includes access to all the academic resources and as well as the content of all other portals. So the academic portal is designated for scientific research and higher education. There are two subtypes of academic accounts on EKB, university and research centers staff and university students. So the requirements for the reader portal is just any email and national ID. Academic has additional requirements, including the university or institutional ID for students, or having an institutional email or access to the institutional internet network or IP range to verify as staff member. So the lower two are for staff members. The first one is for students. So for registration on EKB, once you go to ekb.eg, you click on register, you create an account, you select the portal that you would want to subscribe to. So for scientific research and higher education, so you get the two options I said earlier, research centers and staff members in addition to university students. So for a limited period, because of the lockdowns and a lot of students accessing research resources now, because in universities there are no longer undergraduate exams except for the final year, everybody is doing research now. So access to the university student portal has been made easier. So there aren't much requirements for university students to actually verify being a student, so it should be accessible to you as well. But to identify as a staff member and an academic member on the portal for university and research center staff members, you would need to either be connected to your internet, to your university internet or your research center internet, or what we call the IP range, or to have an academic email. So academic or researcher email. So these are end with edu.eg or sci.eg.gov.eg or .plp.eg. Once you have started the registration, you'll be required to enter basic information: first name, last name, email, and mobile number, national ID, and birthday. So these personal information include the national ID, means that every Egyptian citizen gets one account. You can actually promote your account from being, if you've already created an account as a reader, you can transform it or upgrade it into a researcher account once you have the criteria. That means the researcher account upgrade, for example. The next step, you will need to insert your professional affiliation, whether you are a university student or a researcher. And there is a caption, there is a confirmation code you will put in. You will receive a confirmation email to your email address you specified. It has a one-time password. As you can see on the registration process, you did not insert a password. So you'll receive a password that will be used only once, and you have to use to sign in with this one-time password within 72 hours of registration. If you do not activate your account within 72 hours, then your registration is canceled and you have to register again. And one thing you need to remember, that the entire platform of ekb.eg is only accessible in Egypt. So once you are on EKB, this is how it would look like. And So you can see that EKB has developed an academic search engine that takes the query or the syntax you've put in for the search, and it runs it against all the data or the majority of the databases available on EKB. It's a very powerful tool, but I would strongly recommend it if you're looking for something very specific. But in order to do more, to use full capacities of very specialized searching and indexing tools, you will need to use the tools themselves that EKB provides as well. So what I would recommend is to go to sources that you can see here. It's so to go to sources, select all sources to see the entire full power of EKB sources. There are multiple publishers, multiple service providers. So for example, all Elsevier products or the majority of Elsevier products are listed under Elsevier. However, there are 4 exceptions to this rule. So we have Embase and Reaxys, which are the topics of our, which are the products that will be or the solutions that will be part of our discussion today. So Embase and Reaxys, since they are available for only a limited period till the end of July, so they have separate portals or separate icons on the sources page. Also, Novel, which is an engineering solution, is also limited till August as well. And there is also SciVal that's not listed under Elsevier, although it's an Elsevier solution, because SciVal is only available to academic accounts. Embase and Reaxys are available to you on reader accounts as well. So if I scroll over, so as you can see here, it is, I can also see Reaxys. If I scroll down, one thing you need to know is that all the sources are organized alphabetically. So this is why Elsevier and Embase are next to one another, because they start with an E. If I scroll down, then I can see Reaxys here, and I can also see SciVal, which is accessible only for researcher accounts. Remember that Embase and Reaxys, as well as other Elsevier solutions, are accessible for both reader and academic accounts under the current configuration set by the Egyptian Knowledge Bank. So that would be a quick introduction to how to access the solution. Once you have clicked on Reaxys or Embase, you will see a description for what the solution does. And under the list of products, if you click on Reaxys, it would drive a link that will grant you access to Reaxys. It's very similar to what you would see also on Embase. So same steps, sources, all sources. And I click on Embase, the description, and if I click on this word here, it will take me to Embase. So that's a bit of an introduction on how to access the solutions which are going to be the focus of our presentation today. I will stop sharing my screen, and I'll be leaving the microphone to my colleague, Dr. Andre, who will carry on with the presentation. Thank you so much.
Thank you, Khalid, for very nice introduction how to access the solution, how to access the Reaxys and Embase. And today we will have a quite scientific discussion, and we will discuss mainly 5 strategies for drug repositioning. The first strategy would be focused on using clinical data for drugs as well as biomolecules to find the information about them, the different aspects of how they was used for the therapy of viral bacterial disorders. But this approach is fully applicable for any type of disorders, for cancer, for diabetes, whatever. It's information focused on getting the clinical data. The next one would be on the search of substances reported against relevant viruses in patents and articles. And also we will discuss the Reaxys capabilities which can support you in synthesizing different molecules. The second approach also applicable to almost any diseases like cancer, HIV, etc. The third approach would be about substances which interact with viral and bacterial proteins, As you might understand, it's not applicable for cancer, for example, because in case of cancer, usually we don't have viral if there is no several diseases simultaneously. But the fourth approach, in the fourth approach, we will investigate compounds affecting human proteins involved into the viral life cycle. But if you're dealing with diabetes or with other non-viral or non-bacterial disorders and you are aware about your targets, you can use them instead of the proteins involved into viral life cycle. And also the fifth strategy would be data-driven assessment of safety of drug candidates. This is a unique approach applicable for any molecules, any drug candidates for any diseases. And before we start, I would like to let you know that the additional materials like user guides, Reaxys user guides, Embase user guides, several workflows, including medicinal chemistry workflows, synthetic workflows, and the certificate of attendance are available on the Research Academy website workshop, and you will need to use this code. I will show this slide again at the end of the webinar, so you can benefit from it at the end of the webinar. Also, I would like to inform you that this webinar is presented as a part of Elsevier cooperation with IKB, and as a part of this cooperation, we arranged 5 webinars. One of them already passed about Reaxys. It was about Reaxys supporting research related to food, agriculture, water technologies. The second one, which happened yesterday, it was focused on writing systematic reviews and clinical trials with Embase. You have today's webinar, and next week we will have a very interesting webinar which would be focused on green chemistry and oil and gas downstream, atom efficiency concept, assessment of toxicity of compounds, etc., etc., etc. And the fifth webinar would be again focused on decision making in clinical practice. Also, I would like to present you the Reaxys and Embase landing pages. So how it looks like. As you see, the presentation is fully filled with different links, and the presentation would be also available on the Research Academy, so you can get benefit of these links when you download the presentation. This is how Reaxys landing page looks like. Here you have the link to the IKB resources web page. From there you can download some additional information, fact sheet, so-called about the Reaxys, how many nice substances it contains. So we have introduction, description, some video, and here you can find a quick user guide, video tutorials, and several workflows related to Reaxys, to search substance, to synthesize substance, and to work with medicinal chemistry, like ligand of dopamine receptors. And also here you see the announcement of the webinars, and I hope soon here, by this link you would be able to access the recording of the Reaxys webinar. The same is available on the Embase landing page. So you can find the information about Embase, about user guides for Embase, how to get access to Embase. This is a link. So you can fully get the benefits of the landing page and the webinars which we are prepared. As for now, we almost finished the administrative part, and now we can focus on different strategies for drug repositioning. As a part of the first approach, we will know more about Embase and the application of clinical data for drug repositioning. It's extremely important to have comprehensive data, and you will know why and how Embase can contribute to it. You will even use this tool to generate insights about anti-infective drugs, even if you are not a very deep expert in viral and bacterial disorders, still you can find some useful information. So for example, you are organic chemist and would like to generate some ideas, check them, and when you succeed in it, you can check them with your colleague from pharmacy or medical department. And also you can get from this discussion how to avoid reading of thousands of articles, but get the useful knowledge in several clicks. Feel free to ask questions using the Q&A tab if you have any questions, and I commit to you that we will answer all of them during the webinar, or after the webinar we will contact you separately by email and answer them. So you can ask your questions anytime, not waiting for the part for the finishing of the webinar. Here, with this slide, I want to compare the traditional drug development approach and drug repositioning, which was presented in this article. So as I promised you, you will have a lot of interesting articles and links to them. And the key benefits of drug repositioning that you can seriously shorter development time, you can decrease the costs of the development, at least because the safety profile of the product already known, because it should be approved drug, and the success rate would be higher because at least because of knowing the safety profile, you can present and get insights about good dosage, pharmacokinetics, etc., 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 etc. So, what also to know about drug repositioning, despite all the positive facts about drug repositioning, you need to know that in some countries the intellectual property issue also need to be solved, because the owner of the drug for sure patented, and you need to check how exactly he patented it. And the good news for the participants of today's webinar that our solution, like Reaxys, can help with intellectual property assessment as well. But let's begin from the Embase as we discussed. The key idea why you can leverage the comprehensiveness of Embase is that Embase is a really unique database, because Embase covers more than 3,000 unique journals, and all journals indexed in Medline. In addition to 3,000 of unique journals, in Embase you can find a lot of conference abstracts, which is called gray literature, and it's really complicated to get access to this information from any other source. But here, just from one platform, you can got the really very comprehensive content. Also, it's important to mention that Embase have very nice indexing, and to understand of the value of indexing, let's compare several searches which you can do, for example, in Google. And if you do search in Google for ivadatin, you will get 10 results. For furadatin, you will get 4,000 results, and furadatin with E, you will get several hundred of results. If you will check it right now, maybe the difference, the exact numbers would be a little bit different, because this slide was prepared a couple of years ago, but the approach in general would be the same. And what is the issue of this approach? The issue of this approach is that all these three is exactly the same drug. it's different trade names of the same drug. And it's become clear that none of these three searches give you comprehensive results, because some of them wouldn't be visible. But the taxonomy, which is implemented in Embase, will let you to find 13,000 articles about this drug, because Embase automatically cover all the synonyms of this drug, which have international non-proprietary name, nitrofurantoin. So as you see, there is about 30 synonyms of this drug, and when literature and conferences indexed in Embase, all of them taken into account. So you, because of comprehensive, because of quite wide coverage of journals and conferences, and also because of very great indexing, you can be sure that you will get comprehensive results. And for indexing, Embase use taxonomy, biomedical taxonomy tree, which is called Emtree. It's really have tree view, and today we will discuss how to leverage all advantages of the fact that it have tree, not just the library, but tree. How the index process is happen. Full text of the articles is indexed together by artificial intelligence and human indexer with relevant background. So both of them together analyze the full text of the article and justify the key concepts discussed in the article. And these concepts are added to the as index terms, Embase index terms, in alignment with taxonomy tree, Emtree, biomedical taxonomy tree. So for each article, you can see all the drugs discussed in the article, all diseases discussed in the article, and additional biomedical terms discussed in the article. Articles. For example, adverse drug reaction, complication, additional information, if it was systematic review, if it was meta-analysis, what was the age, what was the mortality, what was the study group, it was human or animals, because Embase also cover a lot of journal with veterinary research. So the approach which we are discussing today is fully applicable to the veterinary infective drugs and other types of the drugs for veterinary. If we talk about fighting with coronavirus or with some diseases, the first question where we can get some ideas to start from. And the approach is very simple. Right now, it's not the case, but for example, in January and February, where the full structure of COVID-19 was not discovered, even small additional information about what is the protein, what is the structure was highly appreciated. And the idea is the following. So if you don't have information about current virus, some novel virus, you can leverage information from similar viruses. And this approach is discussed in several papers. And Embase is exactly the tool which can support you with this. Just exactly because the taxonomy of Embase have tree view and have 96,000 preferred terms, for example, comparing to Mesh, which have only about 13,000. Embase have 400,000 synonyms, but what is extremely important right now, Embase have detailed taxonomy of bacteria and viruses. It have detailed taxonomy of bacterial and viral infections, and a detailed taxonomy of antiviral and antibiotic drugs. So how we can use it? This is the Embase starting page, and we can get insights immediately from the taxonomy tree, Emtree. So let's start from the HIV, for example. We type HIV and we saw the human immunodeficiency infection. And even what we see, Embase contains several subtypes of HIV. HIV-1, HIV-2, and this one, acute immune deficiency syndrome, also have several subtypes, and you can access them easily. So this is tree view structure. So for example, we go in there and see that this term, acquired immunodeficiency syndrome, is connected to the branch human immunodeficiency virus infection, and also it is connected to combined immunodeficiency. What about coronavirus? If we go to coronaviridae, we will see that we have coronaviridae. We have several beta coronaviruses, including beta coronavirus 1, Middle East respiratory syndrome coronavirus, and SARS-related coronaviruses. So, because these viruses are quite similar, we can leverage for the idea generation both approach. We can search for COVID-19, and also we can search all the drugs which was implemented against all beta coronaviruses. But you have taken in mind that after this idea generation stage, you will need to assess this information, because despite all the similarities, the viruses are a little bit different. So what you see and how it works. Now we do the search on beta coronavirus, and if you go to these filters, and we got 19,000 of articles, you can go to this filter diseases, and you will see all diseases mentioned in this pool of articles. For example, acute respiratory syndrome, coronavirus infection, virus pneumonia, Middle East respiratory syndrome. And what is more important? In Embase, we have triple indexing, and with help of triple indexing, you immediately, in couple of clicks, can get all the drugs which was used against particular disease. For example, acute respiratory syndrome, you need to click here details, click drug therapy, deselect all, and you will see all the drugs which is explained there. Let's imagine that we like ribavirin. We just need to click it, click apply, and we immediately see the articles which explain how ribavirin was used against this disease. And you see the first article is systematic review. So when you click to the article from Embase, you can see the title, you can see the abstract, you can see drugs discussed in the article, you can see diseases discussed in the article, and also you can see other terms. As you see, some of them are highlighted by yellow and some of them highlighted by green. This one highlighted by yellow because we use it in the filters. This one highlighted by green because we use it in the original search, and as you see, it was exploded from beta coronavirus. Also, you can see that some of them are highlighted and some of them are not. The highlighted terms means that this is the major focus of the article. For example, in our case, it's lopinavir coronavirus infection, Middle East respiratory syndrome coronavirus, and SARS coronavirus. In addition to that, I advise you to scroll down. Often, if there was any clinical trials discussed in the article, you can see the clinical trial numbers over there. Clinical trial numbers. So you can directly refer to them. What also is possible in this unique interface. The drugs and diseases terms sometimes are clickable. So for example, you can click on the drug terms, lopinavir, and you will see subheadings. It's additional terms which explain what exactly is discussed in the article. And from this description, we see that in this article, lopinavir, we see subheading drug comparison, and we see details about it. So in this article, lopinavir was compared with beta interferon, and lopinavir was compared with remdesivir. What else we see? We see key subheading drug therapy. It means that lopinavir was used against coronavirus disease 90, coronavirus infection, MERS, and servo-acute respiratory syndrome. Also, you can see subheading pharmacology. It means that the pharmacology is discussed in this article. So, for example, the same is applicable in diseases. You click on the disease's name and you see subheading drug therapy, and you can see all the drugs which was discussed in this article. What is the relation of these to the drug repositioning? The idea is the following. That often the drugs have some exact prescriptions, but this type of search can help you to find all aspects of drug application, on label and off label. And if you see some off-label clinical trials or just cases, how the drug was applicable, it can give you additional information if it has sense to use this drug against this disease, and maybe to make bigger research and to go deeper into this topic. Coming back to COVID-19. Right now, COVID-19 is a candidate term, but even despite these candidate terms, you can find a huge amount of information about it. So almost 18,000 of articles focused on COVID-19. What is important in Embase? Because of a real focus on drug therapy, on pharmacology, it covers a lot of clinical trials and randomized clinical trials and meta-analysis and systematic reviews published not only in English journals, but in non-English journals. And if you're interested in high quality information, you can choose filter study type, and you can select what type you are interested in. For example, in our case, we are mainly interested for humans, but for example, for some typical antibiotics, you can see non-human research there. For example, we see it here as well, and it's will connect you to veterinary drugs. But also, for example, there is practice guidelines. Which is like articles which recommend to use some specific drugs against some specific diseases. Also, what is available here? You can find systematic reviews, cohort analysis, meta-analysis, randomized control trials and topics, and a lot of randomized control trials by itself, and a lot of additional interesting information. This slide explains the concept of triple indexing, but we mainly covered it. So drugs and disease have subheadings. For example, to the disease, we have following subheadings: complication, congenital disorder, diagnosis, disease management, disease resistance, drug therapy, etiology, rehabilitation, side effect. It's subheadings for disease. The subheadings for drug is adverse drug reaction, clinical trials, drug combination, drug comparison, drug dose, drug toxicity, and drug therapy. And for example, pharmacoeconomics, pharmacology. And also, in addition to that, we have 47 different routes of administration for some drugs. Also, Embase have very good indexing of medical devices, and even for medical devices, we have subheadings, which is called adverse device effect, clinical trial, device comparison, and device economics. So this is subheading. Some of them are key subheadings, and in case of key subheadings, you can use the triple indexing. It is exactly what we use in the previous example. So one of the key subheadings, you can see all of them here. One of the key subheadings is adverse drug reaction. So in couple of clicks, you can get the list of adverse drug reactions mentioned in the journals in conference. This is the idea of key subheadings. And it's not just some drug reactions discussed in the article and the name of the drug just suddenly appear in this article, but it's manually excerpted from the article and all of them connected to the drugs with which they are indexed. So as we already discussed, we already have this example, for example, for COVID-19 or for severe-acute respiratory syndrome, you can use drug therapy and get all aspects of on-label and off-label therapy. What I have not shown you, what I haven't shown you is this capability. So from here, you can not only filter it, but also you can export all the drugs which is mentioned in this list. So you can easily put it in your Excel sheet or in some other software. Here it is, the list. Also, from Embase, you can export your search results. For this, you just need to click here. You can click export. You can send them to Mendeley. It's free of charge product from Elsevier, which can help you to manage your references to EndNote, to CSV file, or to any other formats. And for example, for CSV file, you even choose what exactly to export: titles, citations, full record, etc. Also, if you want to keep awareness about some research topic you like your results, you can set up the alert for you. So from this search, you just need to click email alert. But the system advises that first, to manage alert, you need to create login for your system. You need to go there and register or login if you have if you already have your Elsevier credentials. So for example, right now I can easily search for tuberculosis, and we can go to different study types. For example, we are interested in animal tuberculosis. That's why we can choose non-human, for example, for veterinary aspects. And after this, so You see how it works. So it's the search number 5 and additional filters is applied there. And right now you can create the email alert for this particular search. You can make the name for this alert, add some comments, select when you would like to get information, and it will send to you automatically. In this slide, I collected several strategies which is applicable, for which you can use Embase to get relevant and comprehensive information related to drugs R&D. So if you would like to search for drugs recommended in clinical practice against some diseases, you can search this disease in quick search or in Emtree. And after that, you need to use a filter, study type, practice guideline. If needed, you can select years. So for example, you would like to compare what was recommended 20 years ago, what was recommended 10 years ago, what is recommended right now. The results for sure would be different. And after that, you can use triple indexing diseases, select required diseases, go to the subheadings, drug therapy subheadings, and export. So we can do it right now on the example of tuberculosis for veterinary diseases. We have 51,000 of articles. We go to the study type and select practice guidelines. When we found our 700 results, we can go the publication years, filter years, but I don't want to do it right now. And what we can do now, we can go to the diseases and go there, drug therapy, and see the drugs which have which was mentioned in practice guidelines for the non-human application. So you see it's isoniazid, rifampicin, etc., etc., etc. So now you know how to make the search in Embase, how to make the search in taxonomy tree, how to use filters, and how to make general application in Embase. Just want to show you something in Embase. So now we go to bovine and you see the deep of the taxonomy tree. So you see that even just bovine have a lot of different subtypes and all of them indexed separately. If we come back to bacterial disorders, we can find Helicobacter, and you see that it's on different branches. From one side, it's spiral and cured bacteria, but from the other side, it belongs to Helicobacteriaceae. So it's the bacteria by itself. We can go to the articles related to this bacteria and find diseases which discussed in the articles, and non-surprisingly, number one is Helicobacter infection, second is gastritis, etc., etc., etc. We can go back to taxonomy tree and try to search Helicobacter infection. You see, it's separate branch and separate terms, and you see different bacterial infections. So, for example, it's about 1 million records related to bacterial infections. By clicking here, you can get information from the filters about all antibiotics, for example. All drugs mentioned with bacterial infection available here, but from disease filters, you can see different the drugs which was used against some particular disorders. For example, tuberculosis, human immunodeficiency virus, staphylococcus, pneumonia. For sure, the different drugs can be used against different bacterial infections. The same you can see and you can find with viruses. So virus infection, there is a lot of subtypes. We can go to RNA virus and go deeper and try to find what is relevant for you. So it's one approach, it's one strategy. The second approach, if you search for safety information of some drugs of or even group of the drugs, you can search all records related to these drugs or drug type, both from Embase, Emtree, or context search. After you can use a study type filter or skip it. And after this, you can get all adverse drug reaction from triple indexing, or even you can find information about drug interactions. So how it looks like. For example, let's go to retrovirus infection. Let's find some drugs over there and try to see if there is some adverse drug reaction. So for antiretrovirus agents in general, we have more than 5,000 articles about adverse drug reaction. We have the list of them. And also we can see 1,000 of articles focused on drug interaction. So where this antiretroviral agent interact with other drugs. How it is interact with other drugs. So now you know at least two strategies to use Embase. The third strategy is explain how to find registered clinical trials of particular drugs. You need to search records related to some drugs. After that, you need to use subheading clinical trial. You can use some filter if necessary. And after that, you can browse documents to find information about clinical trials. How it works. So we starting from some drugs. And to use drug subheadings, I advise you to start like this. This is starting page. You go to the drugs. You go to the drugs subheadings and type something. For example, trastuzumab. It's antineoplastic drugs, and we are interested in clinical trials. That's why we select this subheading clinical trials. In addition, for example, if we are interested in drug interaction or adverse drug reaction, we can select another subheading, and we even can select how they relate with each other. So if we are interested or in clinical trial or in adverse drug reaction, we need to choose or. But if we are interested in adverse drug reaction which was obtained from clinical trials, we need to choose and. Let's try to do like this to make a quite strict search. But the search is strict. At the same time, you will get precise information which would be highly relevant. Otherwise, trastuzumab by itself can easily give you 100,000 of papers or something comparable. But right now, we have only about 2,000 of documents where we have some adverse drug reaction, and for all of them, we have clinical trial numbers. As you see, it's on below of the page, and this is the number from clin trial goals, but also clinical trial numbers available from other sources. For example, from this one, EU Clin Trial Register. Again, it was you can leverage this using the subheading clinical trial. starting page, drugs subheading. Over there, by the way, you can see routes of drug administration. If you're interested in some particular drug administration, it is subheading as well, so you can focus on some of these strategies. The strategy using number 4 is a strategy to search for preclinical or clinical trials of drug combinations. For this, you need to start from one of the drugs. You can use filters or not if needed, and also you need to choose filter drug combination subheading. How it looks like. So again over there with trastuzumab, we go to the filter drugs. We go to trastuzumab, to its details, and we go there, drug combination. So there is 1,500 articles where trastuzumab was used together with some other drugs. Also, you can find useful information from drug comparison to assess which drug is better, drug interaction, drug therapy we are already discussed, and we have 2 more subheadings related to pharmacovigilance. So this strategies is in presentation. Presentation is available on the Research Academy. So after this webinar will be finished, you can easily download it from Research Academy together with your certificate, together with additional guides, and start using and getting benefits from Embase immediately. While we are talking about anti-infective and antibiotic drugs, we need to mention that Embase also covering even antibiotic resistance. And to search information about it, there is 2 approaches. First approach is the antibiotic resistance is a separate branch in Emtree. It's specific one, so you can type in Emtree antibiotic resistance, see different types of resistance, for example, rifampicin resistance, tetracycline resistance, beta-lactam resistance. It's one approach. The second approach, you can find different bacterial infections and use drug resistance subheadings. So this approach can guide you for some specific bacterial infections. It is also applicable, this drug resistance subheadings is available for all diseases, so you can use it for antiviral drug resistance, you can use it for cancer drug resistance, and I think it's valuable to show how it works. So you start from the beginning, starting page, you go to the specific disease search, you need to go to the disease subheading, and here you see drug resistance. So for example, influenza. Let's find information about drug resistance influenza. Or we can try cancer or whatever. So we found 843 articles, and all of them particularly about the influenza drug resistance. This one is oseltamivir resistant influenza by virus in mice. And by a couple of clicks, you see that 5-vipiravir and oseltamivir was used, and they observed drug resistance. Okay, so if you still have any questions regarding Embase, you can ask them using Q&A, and we will answer them to you during the webinar or will contact you separately. The second approach to find information is focused on the substance reported against viruses or bacteria or any other disease in patents and articles. What is the difference? It is not obligatory clinical trials. It can be reported by anyone else, for example, in patents. And to make this search, we need to leverage the value of Reaxys, because Reaxys is a chemical database inside of which several databases is available. One of them is bibliographic database, 60 million records, and also there is a substance database, more than 100 million substances, and what is important, 500 million experimental properties. And the information about usage exactly there in the properties. Also, it's the chemical database which can help you to understand how to synthesize the molecule. And from EKB, the bioactivity database with 37 million experimental bioactivity data points and more than 27,000 different targets also available via EKB website right now. So why it is important? Because from one substance record, you can get huge amount of different information taken from different patents and articles. For example, this is the page for typical antibiotic doxorubicin. And all the information presented here in the doxorubicin page cover more than 39,000 experimental properties, including 32,000 bioactivity data points. As you see, it's really very well detailed excerpted. So it has the original parameter, it has its value, units, the species, the effect of this compound, etc., etc., etc. And Reaxys have more than 500 different properties. So in addition to bioactivity mentioned and application mentioned, it contains a lot of physicochemical data. It contains solubility, it contains liquid-liquid behavior, it contains spectral analysis, etc., 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 which can be used not only to synthesize the molecule, not only predict its pharmacokinetics, but also it will help you to make the formulation and to fill your master file. In this presentation, in the bottom, you often can see these read words, and this is the links for some useful materials. So, for example, this link will explain you how to search physicochemical properties of substances using Reaxys. I encourage you to watch this video to know how to use Reaxys and to leverage the value of the available data. So, in brief, what is the difference between Reaxys and other databases, for example, Google, Web of Science, or whatever? They all, as a result of your search, give you the list of papers. And to get the answer on your question, you need to get the paper, you need to read the paper, and you need to compare the paper. So you need to spend a lot of effort to get the answer. But once you have access to Reaxys, as well as Embase, especially with triple indexing, you can get your answers directly. And also, I want to highlight that Reaxys is applicable not only for organic chemistry or inorganic synthesis, but also it's applicable for different types of research, and some aspects are covered on our webinar. For example, today we talk about drug development. A couple of days ago, we talked about agricultural applications. In a couple of days, we will talk about environment and safety. We already talked about water treatment. We will talk about energy, oil and gas. So Reaxys have wide range of applications, because if you look around yourself, you will see that everything what around us is related to chemistry. It's painting, it's coating, it's drug, it's food. Chemistry is everywhere, so Reaxys can give benefits to all of them. And let's go to our search strategies. Let's go to search strategies. So if you are interested to find the patents, which substance are patented against some specific diseases, or specific application, by the way, for example, corrosion inhibitor, it's also possible to search there. You can use the searchable field use from the Reaxys. And let's go from Embase to the Reaxys. Sorry. This is how Reaxys starting page looks like. This is a place where you can search something. This is a place where you can search, where you can draw your molecular formula. So for example, if you want to find derivatives of aspirin, you can do it. What you need this button from the structure editor. Insert structure from the name. And you got the aspirin. After this, you can edit it however you want. But it's not the scope of our webinars to cover all details of structure editor. I will just highlight you the some which I like so much. First of all, you can use both Marvin.js or ChemDraw.js, whatever you want. In Marvin.js, I like this one. So it's position variation bond. So we can add any group in any of these position to the aspirin, and we even can set that it would be any atom, heteroatom, metal atom, or halogen. Let's use halogen. In addition to that, we can set that we want to have some cyclic group over there. So to this ethyl, we connect cyclic group. And in addition to that, we can use any group or hydrogen over there. So right now, we can search the structure as drawn, or we can search for any substructure, or what is highly related to patents, we can click there and add all related Marcus structures which are mentioned in the patents indexed by Reaxys. Let's do the search. The search query was not extremely smart. That's why we got only few compounds. So we see exactly what we're looking for. We get halogen there, and we get something connected there and something connected there. But let's come back to the search strategy related to patents and application. How to get there? This is query builder. To get to the query builder, on the top, you need to click query builder, and you will see this screen. What is the idea? In Reaxys, there is 500 searchable fields. And if you want to get some particular information, you need to search in some specific search fields. And the query builder developed exactly to give you this opportunity to search in specific search fields. From quick search give you something very fast and like a fast first approach. But if you don't find, or if you don't like your search results, I advise you to use query builder. By myself, I use query builder like maybe in 50 or even 70% of cases when I can try to search something. So for example, for our use case, we need to search field use. It tell us how exactly the substance was used and what is the substance use pattern. So over there, we can type SARS, coronavirus. What I like there, it's that in Reaxys, in any text search string, first of all, you can use semicolon to separate different terms. It will mean that you use it as or. Also, you can use star, which means any combination of words, and symbols. So, for example, this will cover coronavirus infection, coronavirinae, coronaviridae, etc., etc., etc. So this should bring us a lot of results, but it would be searched only in the field use. And we found 2,000 substances which have patents and or articles explaining their relation. So, for example, hydrocortisone. It is mentioned in these patents in the background of coronavirus pneumonia, quartzitol, coronavirus infection, acute respiratory syndrome, even cadmium. It was reported against SARS. So again, this information comes from patents, which is not obligatory clinically tested. So you have to take in mind when you are doing this search that, for example, you can find not only active ingredients, but maybe, for example, you can find the compounds of the drug formula, because in the patents, it's not always explained what exactly the role of the compound. So, for example, you see catechin is have patents infection disease caused by virus belonging to coronavirus family. So it have no sense even to try it because 20 years from this patent is not expired. This is strategy number one. And what I haven't mentioned is the filters. In the filters, you can filter your search result. We have 2,000 substances by structure. So for example, you want to have some phenolic derivatives. And I advise you to put here substructure and limit to, and it can take some time. But now we should see the only compounds which have aromatic ring in their structure. Also, what is available here? You have filters about highest clinical phase. So just to focus on the compounds which was at least clinically tested, you can use it. Again, click limit to. You can see the targets against which the compounds mentioned in our search results work. Here it is. We have 900 substance with aromatic ring, and as you see, it's highlighted by blue. So let's come back to filters. We have different classes, substance classes filter, and often they are very useful to justify the chemophore. For example, it's obvious that once we filter for aromatic ring, all of compounds have 6-member rings, but some of them even have 9 to 80 members in their rings. And also its availability. It's very interesting point because here you can select the compounds which available for purchase, which have available preparation, and which is even approved drug. Or you can remove the substance for which neither preparation, neither purchase is not available. Let's limit to them and see additional capabilities of Reaxys. So we can talk a little bit about getting substances from over there. So this one, this icon, shopping cart, will bring you to the databases of commercial availability. So by clicking here, you will be redirected. Today this one doesn't work. You will redirected to several suppliers and aggregators of different suppliers. And from these suppliers, you can find the information about commercial availability of the substance. Oh, it asked me to install something. So from there, you find this molecule, CAS number available, MSDS available, some properties, available pricing. Here we go. 5 gram cost $40, 25 gram, $47. It's commercial availability. From here, preparation. You can get the different preparation for the compound. As you see, it's contain different chemical reaction, and as a result, you will get the desired compound. Each reaction can have one or even several articles, for example, like this one, and they even can undergo under different conditions. For example, this one goes with hydrogen chloride in ethanol, but this one goes in acetate buffer with some enzymes. So quite different types of the reactions is presented. And here you even can see experimental procedure. Elsevier have legal agreement with several hundred, about 300 if I'm not mistaken, leading journals, and accept experimental procedures from these journals, and also it's accept experimental procedures from patents. So you can understand how to carry out this reaction even without the access to the full text. But full text is also available, and from Reaxys you can first click here and you will go to the publisher website. Here is it, it's loading. So you can see the article on the publisher website. Also, you see information how this paper is cited to assess which method is preferred. By clicking here, you will be redirected to Scopus, and you even can see who exactly is citing this or that article. So here it is, it's Scopus. So you can, for example, find who is citing this article from the Egypt or from any country. Let's see if there is any Egyptian here. No. Okay, so you can be the first who carry out this reaction from Egypt. And also what Reaxys have, so you see some reaction which you can use to get this substance, but also you can use synthesis planner. You need to click over there. You will see this screen. You can use automatic planner. And let's create the retrosynthetic plan. The Reaxys will collect the puzzle for you and automatically create several plans. So quartzitol can be obtained from this and this molecule, but I don't think it has sense. So let's see what else is available. Well, looks like this compound is a typical intermediate for getting quartzitol. Maybe this is natural compound, but also you got the idea. So for each stage, you see the previous stages, and Reaxys combines this puzzle for you. This is stage number 1, and this is the condition for stage number 1 with experimental procedure. This is stage number 2, show condition. You see the condition for the stage number 2. This is stage number 6. You see condition for stage number 6. And again, we started from the query builder, use field, and found compounds which reported to be active against some viral disorders. You can play with it by yourself. It's what I advise to you. You can try neoplasm, cancer, or whatever, and you will find reported compounds. So this is Reaxys strategy number 1. Reaxys strategy number 2 is to use substance effect search field. Again, type effect, go there, and from here you can browse the different effects. For example, let's focus on the scope on our webinar, but just before it, for example, anti-neoplastic. You see a lot of anti-neoplastic agents, so it can be anti-cancer drug. Anti-diabetic agents, 60,000 molecules, and also antibiotic agents. What is the difference with previous search strategy? Let's get the search. In previous search strategy, it was compounds which just mentioned against again some viral or bacterial or whatever disorder. Right now you get substance which have experimental data, because in Reaxys, in the model medicinal chemistry, we have a huge amount of bioactivity, and you can get the access to it by browsing it. So you will see this bioactivity heat data. Just click on it and you will see this data, this table. So you see the parameter which was exactly measured in the article. You see its value, qualitative and quantitative. You see unit. You can see biological species. You can see the action on target. It was inhibitor. You can see the exact target. You even can see the dose and effect. So this information is obtained from the experiments. So this is much more reliable. That's why I advise you to use both strategy. With previous, you can assess if it is patented. With this strategy, you can get access to the experimental information. And also you even can get insights. For example, which targets can be affected for Mycobacterium tuberculosis H37V to have antibiotic effect. The target is isocitrate lyase. So this was strategy number 2. And also it's important to mention that you can use different combination of strategies. For example, I like this example because triazavirin is antiviral drug developed in Russia, and I'm from Russia. We can search combination of the search fields. How to do it? We go to query builder, we add structure. We just need to click here. Also, we can type use, or The second approach, from there we can go to the history and see all previous searches which we have. So we can do like this. We can even combine them with each other. So here I go. We group use field and previous 2000 molecule which was used, and we can use different logical operator. For example, we can use here operator OR, and it's just an example of how you can combine different search fields. And also we agreed to add the structure of triazavirine. Here we go. As you see, there is a difference, capital letter and small letter, and Reaxys advises several options. Let's load this molecule and let's find all substance, not just triazavirine, but also even similar to triazavirine. And click here. Today looks like today it worked with some bugs. I hope we can finish it. Here it is. We found 15 triazavirine derivatives. All of them have documents connecting them to SARS. So it's the patent which explains that it can be used against SARS. Here it is. Okay. Additional information about using structure editor available in this video. And we already have the demo about the synthesis of different compounds. So let's go. We also discussed several strategies how to assess biosynthesize the compound. As we discussed, this icon will help us to assess commercial availability. With this one, you can create retrosynthesis plan. And from here, you can go to the information about reactions and preparation of the compound. Here, the experimental procedure available. If you would like to use specific solvent or catalyst or have high yield, you can use filters. This is the slides about retrosynthetic planning, which we already discussed. And also, I want to explain one interesting use case. So Reaxys even can be used to predict the direction of the reaction. So for example, if you draw something like this, I will explain you what does it mean. It will give you all the reaction of a compound containing aliphatic and aromatic group. So after getting all the reaction, you can assess what kind of reaction undergo in which conditions and how to make your own reaction in the best way. What is this? This is aromatic ring. This is a bond which can be connected to any atom from this ring. If you join recently, you can watch the recording later, and we already discussed how to use it in structure editor. This is G group. It means anything, exactly anything, even connecting to the cycle. And to the G group, we connected carbon group, 2 carbon groups. So this one is exactly the aliphatic hydroxyl group. This is aromatic hydroxyl group. And by this link, we ensure that this wouldn't be, for for example, acid. So we add arrow saying to the system that it should be the chemical reaction, and as a result we will get 4,000 different reactions. This search takes some time, so I wouldn't show you online. In the filters, you see different reaction classes. So you can see that there is substitution reaction, cyclization, oxidation, reduction, and other types. And by filtering them and by browsing them, you can understand in which conditions this or that reaction undergoes, so you can assess in your condition, will it go or not. Okay. And also very interesting thing is atom mapping. It have sense if you would like to show to the Reaxys that you want to make the reaction on some particular item. How to do it? I show you how to use structure editor for this, but wouldn't show you the search because we have quite limited time right now. So we are doing this search as we previously agreed to. Let's add carbon, hydroxyl over there. And for example, we are interested to oxidize only this hydroxyl group. We don't care about other 4,000 reactions. We need to oxidize only this one. We add arrow. We select it and doing Ctrl+C, Ctrl+V. And after that, we need to focus on the bond. You see, I move mouse there, doing right click, and I see the menu related to this bond. You see bond properties. And here you need to select reacting center. So you see this appears there. So right now Reaxys will bring us only the reaction when this bond is reactive center. There is a small mistake. Let's fix this mistake and make the search. While this search is going, we will come back to it later, but I will show it to you. And as a result, you will see less reaction, and it's exactly the reaction where the structure is kept the same and only one bond act as a reaction center. As you see, this was hydroxyl group. Now we have keta group. The same over the hydroxyl keta group. The rest is kept the same. Hydroxyl keta group. So let's go to the substances with interact with some specific particular proteins. It's the big strategy number 2. And as you may probably know, there is some requirements for the ideal antiviral drug or antibiotic drug. The drug should inhibit or block an action of viral enzyme or some process in the viral replication. The drug should not influence normal vital activity of the host organism. The drug shouldn't be toxic or form toxic substances during the metabolism, and drug shouldn't give adverse drug reaction. Right now, we will address the first point, and these 3 points we will try to find on our 5th strategy. This is a slide which is illustrating the mechanism of action of acyclovir against herpes simplex virus. So the idea of acyclovir is that acyclovir have similar structure to natural substrate. It's guanidine nucleoside analog. The only difference that acyclovir is missing this hydroxyl group and this part of the ring, which play crucial role in the viral replication. So when the acyclovir is inside of the cell, it is, what is important, by the herpes thymidine kinase, it's viral protein, it's not host protein. So, and this viral protein is quite selective. Human proteins activity with acyclovir is much lower, and do not go to this reaction. So if you are healthy, the acyclovir wouldn't affect you the same way. But in case you have herpes simplex virus, the herpes thymidine kinase convert acyclovir to acyclovir monophosphate, which is undergo some further transformation by host proteins, and it's go over there. It go to nucleus where it's inhibit viral replication, because this hydrogen bond of the guanidine nucleoside is a part of the creation of the chain, and acyclovir who are having no this hydroxyl bond stop the formation of the chain and even irreversibly inactivate the protein which is related to this. So the soon cell will die and the viral replication will be stopped. You can find more in these papers. But what does it mean for us? For us, it's really good example of good antiviral drug, and we can try to find something the same. Once we know the target, and we already discussed that it could be a viral target, or it could be even, if you're fighting with some particular disease, it could be human target which is involved into the disease, and with Reaxys medicinal chemistry, we can find information and find substances which interact exactly with this target. So if you have some ideas, you can start right now, but even if you don't have ideas, again, you can leverage the taxonomy. It's our friend who will help us, who will support us, because in Reaxys you also have the taxonomy of targets, you have a taxonomy of species, and it's like key points which we will leverage. In this slide, you see all the searchable fields where taxonomy is available. The availability of taxonomy you can assess by this icon. And today we will use biology species or target name. How it can help us? The idea is very simple. So you go to query builder, you search for biological species, searchable field, type it, and go to the taxonomy. In the taxonomy, you can find the species, viral or bacteria against which you are interested in, or you even can go one level up or even two level ups and find all family to try to develop some molecules against whole family, because first of all, maybe the molecule is not just yet tested against particularly your virus, but also the proteins in the family can be the same, and this is already explained in several papers. So for example, COVID-19 have similar proteins than SARS, for example. And the same approach can be used with target names. How exactly it's related to target? The target name have the name of the proteins, but also in the bracket the target name contain the information about to which species it belongs. So you can use both of them, and I advise you to use both of them because unfortunately sometimes there is issue with indexing. For example, in the paper it's unclear to which to which species this target belong, or what was the target name. So it's to get comprehensive results, it's better to use both of them. From papers, we already know several well-known targets identified to fight with coronavirus. And you can find some information on the slide, but if you browse all the articles, potentially you can find even more. But let's do our search. Let's see. As I promised to you, Reaxys bring us the result where we can see that hydroxyl group was oxidized to keto group. But right now, let's focus on our species. Biological species and target. I advise you for this particular strategy use OR. For example, we are interested in coronavirus. Let's select all coronavirinae. And here it works a little bit differently there. So you can see, you can search for some class, but also you can search in the name of the protein. So with target name, it will have sense to mention something more. So for example, like SARS, MERS, to add there. In the name of biological species, it have no sense to add them. Okay, first of all, let's find all targets related to coronavirus. And we found 68 targets. And as you see, it's different species, mutated, wild, whatever. And after that, just in one click, you need to click there, substances. You will get 2,000 substances which have reported bioactivity against this target. What else is important? For example, let's go to the quercetal. If you go over there, you will see that it's often happen, the units is really different in different papers. So for example, this author use milligram per milliliter. This author use micromole. And directly to compare this 1.7 and 8.1, it's a little bit complicated. What you can do, you can leverage the Reaxys, because Reaxys medicinal chemistry convert this value into the unitless size and make logarithm, and the px is exactly the logarithm of the unit expressed in the paper. So here you directly can compare and see that it's more or less the same bioactivity. Why I explain it to you right now? Because we have 2,000 compounds which have some reported experiments, but we are interested in only highly active. So what we can do is to filter them to the substances having high px, more than 5 or 6, it means that bioactive concentration would be less than -5, 10 in -5 -6. So here it is. In a couple of clicks, we have 800 candidates for the drug repurposing against COVID-19. You can click here and find that 16 of them already market approved drugs. Several of them have phase 2. Here it is. So this is how you can leverage experimental data for generating ideas for drug repurposing. What else is interesting there and what you need to know is this heat map. So with this heat map, you can put into one sheet all bioactivity and for all substances. So in this case, you will have the axis X with substances, or you can change it, but I wouldn't. Also, you can use axis with target or with biological species, but let's keep target. And you will see all the target and all the substances. As you remember, we filtered it previously. But right now, you see this target, DNA SARS, papain-like protease MERS, papain-like protease SARS, etc., etc., etc. This one, replicase polyprotein 1AB, and other well-known targets. So with this, you can assess your bioactivity. And we will come back to this tool when we will assess the safety of compounds, because right now we are limited only to viral targets and viral bioactivity, and so there we see only viral proteins. What also is interesting, from any stage you can export data. What you need is just to click export button. You can select format, you can select all results or something specific, and you can select what exactly you want to export. All data, heat data, or some specific data. For example, you can export only bioactivity for coronavirus related proteins for these substances. Or you can go back to 800 proteins and limit information for that substances. Often people ask how it looks like, and in export it looks following. So this is structure. This is information about the molecule, the stage. Here you see the name of the target, helicase. Here you see substance actions on the target, the parameter which was measured, IC50, the units of the parameter, and the value, more than 50. And the same for all the rest data points. So you see in a couple of clicks, we analyzed 150 data points. And also PX over there is available. So you can sort them by bioactivity. For example, this one, this number 1 means that the bioactivity is quite low. Measured at PX. Okay. What else we can do? What else we can do? What else we can do? We can assess the other potential molecules. So we already discussed this workflow, and for example, we like quercetol and want to assess bioactivity of quercetol chemophore. What we need to do? Right now, we see the only bioactivity of viral proteins, but we want to have heat map of all proteins, including human proteins. How we can get this information? From over there, from quercetol, we go there and copy structure to query. It appears over there, and we select here all substructure. It means all derivatives of quercetol. Transfer to query, make the search, and we will find 30,000 derivatives of quercetol, which is quite a big target. After that, let's limit only. Let's go to the heat map. Let's go to the heat map. Previously, let's limit only high bioactivity to make the system working faster. So with high bioactivity, we have only 800 compounds, and now we can go to the heat map. Over there, you see now huge amount of targets. And as you see, for example, this one, 20S proteasome human. This is all available targets in database. Viral targets, bacterial targets, rat targets, human targets, whatever. If we want to assess only to the human targets, we can select only human targets. Click here, target species, limit to. And with this, right now, we have only human targets. And you see these rings. It means the amount of data available for this target. So people often test aldose reductase, and from over there you can even sort which compounds have high activity for against this target. So you see, with aldose reductase, there is 1, 2, 3, 4, 5, 6, 7, 8. 8 molecules was tested against it. And by assessing the structure, you can connect the structure activity and activity if there is some logic. If there is some logic. So you see that this molecule have activity 1-fold higher than this one. And here you can find all different types of targets. So for example, this target, Aurora kinase, is related to cancer and often used as anti-cancer target. Probably you can find some targets related to the adverse drug reactions, for cardiac adverse drug reaction, for toxicity. By the way, coming to the toxicity, from here, you can go to setting, and instead of target, you can choose effect. And from effect, you see it's not only antiviral effect, but also you see here cytotoxicity. So you can sort your molecules by the molecules with high cytotoxicity. Fortunately, coming to quercetol, we don't see serious cytotoxicity, but you can use this tool to assess other molecules. You see some of them even was tested on hepatotoxicity, and yeah, quercetol also have it, and I cannot say that it's low. Okay, so let's come back to the presentation. We already discussed that you can make structure-activity relation by typing, even in quick search, some scaffold, search for example, similar compounds or derivatives. After that, you can go to the heat map and assess all the bioactivity related to this scaffold. The same approach could be used, more or less the same approach could be used if you're interested to some proteins affecting, some molecules affecting human proteins. And often there is pros and cons when you develop an antiviral and antibiotic drugs. So, for example, using some compounds which affecting host proteins can help you to overcome drug resistance. It can help you to cover broad spectrum of viruses or bacteria, but for sure it may lead for some side effects. And if we talk about targeting human proteins, the workflow in Reaxys is almost the same, but I want to highlight and put your attention into one of the process which is highly related, for example, to coronavirus. And this process is called autophagy. And by this link, we have another solution which is called Pathway Studio. And from Pathway Studio, we accepted 400 compounds which inhibiting autophagy and 800 compounds which activating autophagy. So these compounds should be available by these links. And you can download them freely from the Elsevier website. And last but not least is the assessment of the safety of the drug candidate. So it's very interesting use case, and we will come back to Embase, and also we will talk a little bit about Reaxys as well. In Embase, for example, if you will start from Streptococcus pneumoniae, you can find 6,000 papers. You can find 6,000 papers, and by limiting even to humans, by triple indexing drug therapy, you can find different drugs. Most of them are antibiotic drugs, but some of them are methylprednisolone, prednisolone, and prednisone. And it's a little bit strange to see that they used against pneumonia, because glucocorticoids are often used more against inflammation. But here you see it was used, at least they was used together with other drugs. But we got this insight, and let's assess the idea. Let's assess the idea of using prednisone. And with the database, with Embase, we can assess the side effects of the drugs. I will remind you. So you start from the first screen. You type the name of the drug. It automatically connects synonyms and search really very broadly. We found 200,000, around 200,000 articles. And from here, we see prednisone, and we can get the list of adverse drug reaction of prednisone. So let's wait a little bit because it's audit. You would be really surprised, because you can see that pneumonia by itself is a quite common adverse drug reaction of prednisone by itself. So it maybe sounds a little bit strange when you're trying to treat the disease, which can be side effect of the drug which you are using. And we can go into the details. We can find these 200 drugs, 200 articles where pneumonia was a side effect of the prednisone, and try to analyze what's going on there. And if you go to these articles, we can see with subheading drug therapy against which diseases prednisone was used there. And as you see, here the prednisone was mainly used against some lymphomas. So maybe it have sense and it have logic to use prednisone even against pneumonia, but you should be careful if the patient have some myelomas or tumors. What is another approach? What is another approach? Is to assess drug interaction. Drug interaction, and it's quite easy. Again, it's triple indexing. So you can even use very interesting search. Very interesting search. For example, how to assess drug interaction of prednisone with all antibiotics. What is the issue? I will explain you. Here, you see 200,000 articles. If you just open triple indexing, and open prednisone drug interaction, you will see 600 articles and different drugs. For example, tacrolimus, definitely it's not antibiotic. Rifampicin, it's antibiotic. And if you are not expert if it is antibiotic or not, or if you are lazy like me, you can do some other search strategy. And I will show it very nice search strategy which can be which can help you in this case. So what we will do, we will use Emtree. We will first of all find all antibiotic agents, transfer them to our query, use drug subheadings, and select this drug interaction. It's our search query. 25,000. Our second query would be to find all prednisone drug interaction. Prednisone drug interaction. We found much less, 600 papers, and now we can overlap them. So in each article, which we should get in the result, it's only 250 articles, each of them have to contain some information about drug interaction of prednisone and drug interaction of antibiotics. In this case, the probability to see antibiotic there is much higher, and you can expect that at least you do your best to avoid the non-antibiotic drugs. I hope you got the idea. Also, it's very interesting. It's very interesting because when assessing the drug, you should avoid the drugs which cause the symptoms of the disease. And one of well-known symptoms of COVID-19 is anosmia. And you can use triple indexing to find all the drugs which cause anosmia as a side effect. So we have this key subheading. First of all, you search anosmia. After that, you go to the disease filter. choose anosmia, choose side effect, and assess the drugs. By the way, prednisone is also there. So prednisone often causes anosmia, so I assume that it's not the best drug, or at least it should be used with a lot of attention if it would be used to prevent of COVID-19. What else you can do? We can come back to the Reaxys, and we already almost discussed this workflow. So you can make the search of some chemophore. You can draw it by yourself or extract from your search results. After that, so the key point, you have to start from the beginning, from the search. You have to start from the chemophore. And when you draw this molecule, you should search all derivatives of this molecule. I will remind you. So to search derivatives, you need to click the search as substructure or even similar. After that, you will get all substances. You go to view results. In filters, you select human, or due to the similarity, you can add rat and mouse. You select only high activity, and you will get the all information about different affected related targets. So by assessing this target, you can predict potential safety and adverse drug reaction of this or that chemophore. What are the options? What is other option? What is other option? The other option is to try to find some information where it's exactly mentioned. So if you're interested in particularly in toxicity, not in general in adverse drug reaction, you can use a specific form. First of all, it's toxic substance effect, and also its specific bioassays, which covered by Reaxys medicinal chemistry. And let's see how you can do it. Here we go. Quercetal them. Let's remove some group to search on the important part of the chemophore. We search substance effect there, and also bioassay. Bioassay category. So substance effect, we can browse it. You see there is different types of toxicity, also some cytotoxicity available. So my suggestion is to put contain toxic to cover all of them, and also we can select some specific bioassay. Let's select toxicity on safety pharmacology. So here we put or, here we put and, because we want to that all of them would be this contain this chemophore, and this would be the toxicity and safety data. And let's go to the substance. So we got 6,000 substances, but for each of them, you can see this bioactivity heat data, and over there exactly would be toxicity, safety, pharmacology, some research related to it, or some information where it was found that the compound is toxic, cytotoxic, and something like that. So you see different qualitative data and quantitative data as well. So for now, we almost covered all the materials, all the materials which I have prepared for you. I remind you that the presentation and additional materials like several workflows, guidelines on Reaxys and Embase available on the Research Academy. You need to go this link and you will need to use this code. My advice to you is right now to make a picture of the screen by keyboard, by computer, or just by phone to remember the code and to remember the link where you can download the presentation and additional materials. And I also want to remind you about the webinars. The link to the registration of the webinars available on the Reaxys web landing pages, this one or this one, and I will bring you back this slide, and let's go to the questions. Let's go to the questions. I hope my colleagues can help us with the questions.
Andrei, there is a question. Is Reaxys includes the theoretical data such as HOMA and LUMA of published compound? Are we able to answer?
I'm sorry, but I do not understand what is HOMA and LUMA. We will try to understand the question better, and we'll come back to you by by the email. Any other questions?
A lot of very specific questions.
But even if they are specific, can you raise them?
Mm-hmm. Difficult or not habituate to use. Okay. I think it's really very specific.
Khalid, can you help Alexei with the questions, raising the questions?
Okay, so there are I see there's one general case question. So are the solutions you're presenting today restricted to drug research?
Crop research or
drug?
No. It's a very interesting and good point. Previously, 2 days ago, we explain how this solution can be used, for example, for agricultural research, because also Reaxys medicinal chemistry really cover the targets from plants, from animals, and you can you can use it as well. So, for example, you can start from some plant target or from a plant disease and develop some pesticides, herbicides, plant protective molecules, etc., etc., etc. So it's indeed not limited to the human drugs. Also, it's applicable for the plants, for veterinary drugs, and different other aspects.
And also, I believe, if I may add, so we've been showcasing through the webinar series uses for Reaxys, Embase, and Reaxys medicinal chemistry for a variety of applications. So, for example, the day before, on Tuesday, we had a webinar that was related to agriculture, right? And water. So there are applications that can relate to water, environment, so not necessarily drug research, but that was the scope of today's webinar, right?
Yes, definitely. If you are interested in environment research, in safety research, in green chemistry, we are welcome you to our ongoing webinar, which will happen next week.
So there are very topic related or specific questions that might be related to a specific topic or a specific research and that we are receiving.
Maybe we can
And
discuss some of them.
yeah. So I believe already Alexei has been replying in writing for a lot of the questions that were asked, even some on specific cases. So people should be able to see answered questions already on Q&A, just not to take the entire webinar questions and answers into a very specific topic for a very specific research, but I do believe that both of you, Andre and Alexei, you can actually get back to the questions being asked, right? So
Yes,
if they are covering a
definitely.
very specific topic.
Yes, definitely. We will answer the questions directly by email to the participants.
There is one other question. Can the data map of queries released be downloaded in the form of a figure or even a file to present?
You mean heat map? I don't think that heat map can be downloaded in the graphical form, but once you download the PX value, you easily can use Excel capabilities like a pivot table or some graphical tools to visualize it. The reason why it is not implemented because there is so huge amount of different ideas how to visualize the data, so it's impossible to implement it all of them. But one of them, which is also very important, is implemented in the Reaxys, and this is drug likeness. So as pharmacologists know, there is Lipinski's rules and Weber rules, which have some quantitative predictors. And for each molecules in Reaxys, you can open this drug likeness and assess if the molecule looks like a drug. So if it is the molecule, all the points of the molecule should be inside of this, let's call it triangle. If they go outside, most likely this wouldn't be drug-like molecule. Any other question? Do you see any similar questions? Maybe if there is several similar questions, we can address them.
Yeah, so there are some, we have received multiple questions regarding the recording for the session. Some people are expressing there was a lot of features, a lot of steps that were included in the webinar. So if you need to revisit the recording, there are actually two ways you can revisit the recording. One of them is because this webinar is being streamed live on our Facebook page, Elsevier Africa. If you go to Elsevier Africa page on Facebook, facebook.com/Elsevier Africa, just without spaces, you can find the live streaming happening now, and it will be available automatically as a recording afterwards. And please know that the videos from all the official recordings for all our webinars are posted periodically on our YouTube channel. So I will type in the link in the chat for both the YouTube channel as well as our Facebook page. There was a question.
And another option. I hope soon the recordings of the webinar would be available on the Reaxys landing page, which is right now you can see on the screen, elsevierafrica.org/Reaxys/. Yes, there was some other common question.
There is also a question about the upcoming webinars as well.
The upcoming webinars will happen 16th of June. And the name of the webinar is how Reaxys can increase the efficiency of research related to energy and environment pillars. I think it was already discussed and announced. And another next webinar will happen the next week as well, but it would be focused on the Embase. So this webinar will happen 17th of June, and it will be focused on high quality systematic reviews and innovative treatment. So links to the registration available from the Reaxys landing page. What I see is that Khalid is put the links to YouTube channel, Facebook channel for Elsevier Africa group, where the recordings would be available, and the landing pages as well. So everybody who are still with us are able to go to the chat and copy these links. I think we can keep for a couple of minutes and to give a chance to everybody to copy these links, and maybe we can answer some other questions online.
The role of anti-IL-6 in treatment of critical stage of cytokine storm of COVID-19 cases.
Well, I think it's really very specific case, and I advise the author to try to find it in Embase by himself, but for sure he can use the Embase to do it. Also, what I see, there is questions about the difference of Reaxys with other databases. And if there is such questions, we can come back to this slide, and briefly explain the difference. So there is only one common database which contain literature, chemical reaction, and substances. But unfortunately, which is not Reaxys for sure, but unfortunately, this database do not contain almost any experimental properties of the compound. The amount of experimental properties in this database is several times lower than in Reaxys. In Reaxys, we have 500 million experimental properties, but in that other database, there is only like 5 or 6 experimental properties, and mainly they are very simple, like melting point, boiling point. But at the same time, this database have a lot of prophetic data, which you even can predict by yourself using ICD labs. If to compare with the databases about bioactivity, there is 2 types of databases. There is freely available databases with bioactivity, and usually if such databases cover several thousands of molecules, it's already very good publicly available databases. The commercial available databases of bioactivity still cover much less experimental data, much less experimental data than Reaxys. And in best scenario, if I'm not mistaken, it covers like 15 million. And not all of them are, like for example, Chambal, for example, do not cover experimental data from patents, only from journals, And you lack serious amount of bioactivities, because pharmaceutical company who make main contribution, main part into the bioactivity research, they usually do not publish in journals, they publish patents, they protect their intellectual properties. Also, I see the question about the prediction capabilities of the Reaxys. It's on the development stage. Very soon Reaxys will have prediction capabilities, and also it will be able to predict even the synthesis of unknown reaction. It will be able to predict bioactivity, but also even right now, Reaxys cover huge amount of types of bioactivity. For example, it cover even the pharmacokinetic data. So to access pharmacokinetic data, you just need to go to the bioactivity all and find the pharmacokinetic data, which is experimental and excerpted from the literature. So right now, Egyptian customers are able to get benefits of access to experimental data from the Reaxys, particularly pharmacokinetics, like this one, or for example, for solubility data, which is also available in the properties, in other properties. Here it is, solubility. Any other questions?
Andre, can we go over again to the slide where people, to show how people can get their certificates and how they can download the presentation materials, please?
Okay. I see still there is a lot of questions, but we need to stop for the moment, and we will try to answer the questions by directly to the question authors by email. And feel free to visit our webinars, and feel free to get your certificate on the Research Academy webpage. And also on the Reaxys landing page, you can subscribe yourself to the additional information related to Reaxys. And with all of this, I highly appreciate everybody who visit our webinar. I hope you can benefit from some information you got on the webinar. I hope you can benefit from the Reaxys. And with this, I'm stop sharing and wish you the best and good luck.